So today I'm going to tell you a little bit about a story that we've just started working on. So there's a lot of preliminary data in this presentation. And then towards the end, there's some work that we've been doing with Susan Moore at the NIH, and that's just been submitted. So hopefully it'll be a general talk, but also I'll try to go into depth where I can. So first, I'd like to start off and say, well, thank you very much for inviting me to Sao Paulo. It's nice and warm here, because we've had a bloody awful winter in the US. We've got this thing called the polar vortex that's been ravaging most of the United States, and so we've been having snow. And this is a picture from my front window. And believe it or not, that black blob is my car, which I had to dig out. And this is sometime at the beginning of March I took this. So it's yeah, it's been pretty bad. So coming here into 88 degrees is absolutely fantastic. Okay, so what I'm going to talk about today is we're going to try and go through defining B-cell dysfunction in HIV, and really I think this has some implications for vaccine design. So I'm going to give you a brief introduction. Then I'll define some what we now know as aberrant B-cell subsets in HIV infection. I'll go on and show you a study that we've been doing on some ART treatments, and then some gene expression profiles. And in previous years, if you've attended the course, you'll know that one of the tools that we use at the Vaccine Research Center is the Fluidigm technology, which is a multiplexed real-time PCR platform. And then finally, some work that we've been doing with Susan Moore and Dr. Fauci, which is looking at HI-specific B cells during HIV infection. So first off, I'm going to apologize for this very simplistic version of how B cells actually meet their cognate antigen. So essentially this is in the light zone. DC picks up so dendritic cell picks up antigen and presents it to a CD4 T cell, which in turn gives help to a naive B cell that has a BCR that is specific for that antigen. The naive B cell then moves into the dark zone, where it undergoes proliferation and somatic hypermutation. SHM is something that you're going to hear a lot about, not only at this meeting, but any other meeting right now, because of its importance in HIV vaccine research. So we don't know what the signal is, but eventually the B cell can move back into the light zone, get more help from CD4 T cells, and this can go on a number of times. But we don't actually know what the signal is that would move the B cell out of the lymph node and into the periphery, where it starts doing its job of secreting antibodies with effector functions. So if you're interested in this, see reviews by Jason Sister, and also two very, very smart Brazilian people, Gabriel Victoria and Michel Nussensweig. So if you need to know more about the CD4 T cells, I think Rick Kaup's going to address this on Monday. So he's got a very interesting story looking at the CD4 T cells within the lymph node. So an interim goal for an HIV vaccine that elicits functional antibody may be to control infection. However, in HIV, it swiftly impairs the B cell responses. So HIV swiftly impairs the B cell responses indirectly via a CD4 depletion, via polyclonal activation, by bacterial translocation. I'm almost contracted to say something about bacterial translocation but Danny's not here, is he yet? So that's fine. So then there's a reduction of peripheral, naive, and resting memory B-cell memory pools. Now the resting memory pool, as I'll emphasize through this, is where your recall antigen resides. So if you've been vaccinated, those antigen-specific B cells reside in the resting memory pool. Another feature of HIV is that there's poor responses to recall antigen and to vaccinations such as pneumococcus, influenza, and hepatitis B. Thank you very much. However, antiviral antiretroviral therapy restores most of these B cell functions, particularly when started early, and we have some data to show that. So let's see about defining some of the subsets. Again, this is a very stylized diagram where we have long-lived plasma cells that reside in the bone marrow, and this is the phenotype that you'll find in the bone marrow of those. And also immature B cells reside in the bone marrow. They traffic into the periphery where they eventually move to the lymph node, encounter antigen, and then we can find activated mature B cells, we can find a population of exhausted B cells, and also resting memory B cells. In HIV infection, we have an increase in this translational population that's associated with T-cell lymphopenia. We have an increase in the activated and exhausted populations, which is associated with HIV viremia. And also, as I've just mentioned, you have a decrease in this resting memory pool, which has implications for a partially effective HIV vaccine or any other vaccine. So let's just consider this if we're at the flow cytometer. So what this is a flow plot of CD21 and CD27. This is an HIV negative individual, and this is an HIV positive individual. Straight away you can see that these are not equal, because we have a large population of tissue-like memory or exhausted B cells, and this is and they have increased levels of inhibitory receptors like FcRl4, altered expression of homing receptors, stunted somatic hypermutation and replication, and enriched in HIV-specific B cell responses are here. And I'll go on and show you some of that towards the end of the talk. And these generally arise and continue to arise in chronic viremia. Then we have the activated memory population, which is also elevated compared to HIV-negative individuals. And these have increased levels of Ki67. Within this population, you can also find some plasmablasts as well. You can stain, so if you gate on this population, actually stain for plasmablasts. These are very prone to fast-mediated apoptosis. They've got a high level of CD95. They're associated with HIV-induced immune activation as well. Finally, you have this resting memory population, which is reduced in HIV-infected individuals. And this is critical for maintaining humoral immunity, as I say, to recall antigens. So this is what it would look like. Adam Wheatley in the group, he took HIV negative individuals, HIV positive individuals, and a few individuals that we could find that were on ART. And he looked at the naive. Basically, he did the same stain, and this just describes the dysfunction that we find in HIV positive individuals. So we can see that there's a reduction in naive cells. We didn't really find the big reduction in the resting memory population that we should find, but it's well documented in the literature. There's certainly an increase in the tissue-like or exhausted memory population, and there's an increase in the activated memory population. So the CD21 negative populations are really perturbed in HIV infection. So this is a slide I borrowed from Susan Moore, and this is a model for B-cell exhaustion, where we have naive cells, and then we have response to antigen, and in general we have resolved infection would lead to arresting memory B-cell pool, and also long-lived plasma cells. However, with persistent infection, you get this rise of exhausted B cells, activated B cells, and short-lived plasmablasts. And these are some of the properties that I've mentioned already: poor proliferative response enriched in virus-specific responses. This also actually happens in malaria as well. So we're trying to find cohorts in malaria-infected people, and also we're trying to extend that into co-infected individuals, HIV and malaria co-infected individuals. So what's the effects of ART treatment interruption on B cells? So as I've said, antiretroviral therapy restores humoral responses to common vaccines. This is well documented for influenza, for pneumococcus, etc., and hepatitis B. However, if we discontinue therapy with interruption, what is the effect of B cell subsets associated with dysfunction compared to no treatment at all? And this is a collaboration that we formed with Godelieve Debris from the University of Amsterdam, who came to the lab to perform these experiments. So this was the study, and data from the Primo study showed that early treatment of acute HIV-infected individuals have a significantly lower viral set point after treatment interruption when compared to untreated individuals. So essentially what we're doing, we're comparing this line of individuals who have not been treated with treatment interruption. So individuals in these two groups have been treated with antiretroviral therapy for either 6 months or 60 weeks. Clearly this is a moving target because they recruited 33, 38 and 38 individuals into this group. And as their CD4 counts drop, they then get put back on therapy. And so what this is the numbers remaining in each group as we move towards the 144. And this is actually published in PLOS Medicine. So we took some individuals at 36 weeks, which is the virus set point, and at 77 weeks, which is sometime down the road. And we looked at the untreated individuals and treated. I've actually put this in inverted commas because some of this group actually began to receive antiretroviral therapy due to the fact that their CD4 counts were dropping. So this is just a generalized picture of what's going on in the study. So the naive pool remains impaired. Circulating plasmablasts here decrease with, so you have a recovery of your plasmablasts in this system. Resting memory recovers with antiretroviral therapy. And also the tissue-like or exhausted B cells remain in untreated. This is a constant presence in HIV infection, this exhausted population. So what we can conclude from this is unabated HIV replication promotes the accumulation of B cell subsets associated with dysfunction. Untreated patients also demonstrate a significant increase in the HIV GP120 antibodies. I didn't show that data, compared to those who received 6 months of therapy. And there was no difference in neutralization breadth in this study. So what we're doing with this study is that we're breaking the data down by time off therapy, so that will complicate these plots entirely. And then we're looking at gene expression profiles associated with interruption in the untreated individuals. So we've done some initial studies of B-cell gene expression profiles in infection. So Adam Wheatley in the lab has developed a panel that can identify 15 different types of, or 50 different phenotypes of B cells in circulation at any one time. He's done this on a significant number of individuals now, so this is very much repeated, and this is the gating structure that he uses. So it's just a table summarizing all the different subtypes, and these are the frequencies. These populations name change for whatever paper that you're reading, because everyone's got their own name for these. So basically, just have a look at these markers. What he also then went on and did with Sandeep Nepala and Kipper in the lab was that he took around about 250 genes and performed a multiplexed real-time PCR with those genes. And they whittled it down until we found 96 genes that can actually identify each of the different phenotypes. And so even if we didn't do phenotyping and he just gave us the RNA, we could tell you what the phenotype of the B cells is in the preparation just by doing Fluidigm in this system. So these are the genes which clearly you can't see, but these are the genes that are associated with each of the 15 different phenotypes that we find in this table. So this is quite a powerful technique. If we do a principal component analysis on just a few of these genes, we can actually separate out the B cell memories and the natural effectors from the IgA activated tissue B cells that are in the periphery, and also the naive. So this is a very powerful technique. So then what Adam did, he then went on and he took 5 healthy controls, 5 HIV positive untreated individuals, and 5 individuals that were on active retroviral therapy. And he performed Fluidigm analysis at 100 cells per well. So we sorted 3 populations because of the frequency of the populations in the cohort that we had. Naive, decreased in HIV positive. IgG memory B cells, these are decreased. This is essentially a resting memory pool. These are decreased in HIV positive. Then the CD21 negatives, which I've already shown you, are increased in HIV individuals. And I'm actually showing probably more heat maps than than Rafik did yesterday. So, so as we can see, this is the result, and there is difference across the naive B-cell memory and CD21, the CD21 negative cells, as we would expect, because they're different phenotypes. But also, there are differences here within the HIV, those individuals that are not on therapy and those individuals that are off therapy. So we've just started to analyze a lot of this data, and so there are many transcriptional changes associated with HIV infection in each of these populations. So the first analysis highlights, and this is just a representation, that is the reduced homing markers, such as CD21, which we know because it's a CD21 negative population, CCR7, which is important for homing to the lymph node, and CXCR5, which is important once you get to the lymph node. There's an increase in CD69, CD86, and IL-6 retroactivation markers. There's clearly an increased expression of inhibitors, of BCR inhibitors. This is the FCRL family, which are associated with exhaustion. So if you want to include a marker in your panels, FCRL4 positive cells are the ones that mark the exhausted cells. And also this other inhibitor, which is CD22. So the next steps in this is to increase the sample number and perform some flow cytometry on the markers that we find to confirm the RNA to protein. So we found what the RNA signature is. Can we actually relate that by going back and doing staining again on these individuals? And also investigate HIV-specific B cells in the lymph node as well. And we're actually doing that pretty soon, actually. So what phenotypes of HIV-specific B cells predominate during infection? So what I'm going to do here is actually show you how we isolate HIV-specific B cells to obtain neutralizing antibodies, which is how do we mark B cells that are specific for HIV proteins, and what's the phenotype of HIV-specific B cells during infection. This is schematic. Again, I pinched this one from Susan. This is schematic of a GP140, and the trick is here is that we make all our proteins with an avi-tag, so we can then directly conjugate these via streptavidin to a fluorochrome. We can do this for GP120, we've got constructs for GP41, we've got scaffolded constructs for V1, V2 of different clades, and also now we're getting scaffolded constructs of V3. So this is a very powerful technique. This is just an example of some staining that we've done with a HIV consensus B GP140. It's a CD19 positive population, so these are all B cells, and then if you have a look at our friends, the CD21 and the CD27, we see that we can actually define in this individual who's actually on therapy, we can define the tissue-like memory, the activated, the resting memory, which is recovering in this individual, and the naive. Then we can also have a look at the IgG, when these cells are predominantly IgG, the B cells that we can pull out with the GP140. We actually pull out some naive B cells as well, which is kind of interesting, and that's been followed up with David Montefiore right now. So this is, again, another schematic showing the strategy to isolate and characterize HIV-specific antibodies from infected individuals. First off, you have to do a neutralization assay to find out if the individual that you're looking at actually does have any neutralizing antibodies. You can then sequence the whole of the B-cell repertoire and then pull out sequences that you predict will bind to HIV. Or you can use probe-based techniques. This is how we found VRC01 by using a mutant VRC01, which is mutated in the CD4 binding region. You can do direct analysis. We've actually been doing this for flu, where you can just pull out your probe of choice and your CD20, and then you can go on and you can sort single cells and do just plain old Sanger sequencing. Or you can actually culture the B cells, which has been done by in a recent paper with Penny Moore by Nicole Daré-Rose in the BRC as well. So all these will identify and pull out neutralizing antibodies, and then we can have a look at what the heavy chain and light chain is, etc., etc. And this has been done for a lot of antibodies now. So this is a diagram from a review from Peter Kwong and John Muscola. And it highlights the 4 areas of vulnerability on the HIV spike. So EMPA, and we have a number of antibodies that have been identified to EMPA, V1, V2, the glycan V3 site, and the CD4 binding site. Here we have VRCO1, which I'll come back to in a second. One feature of neutralizing antibodies is the high rate of somatic hypermutation that we find in these antibodies. And what this shows is the percentage mutation that we find. VRCO1 is up to 32% mutated. And the way that we work this out is that we have a look at the progenitor, and then we can calculate how many changes and sequence in the CDR3 region. So there's 32% of the CDR3 region being changed to obtain this antibody. And likewise for the other antibodies against HIV. Now, if we compare that to a regular influenza vaccine, we're nowhere near this level of mutation. And also, if you take some individuals and you have a look in the relatively successful RV144 and the phase 1, the RV135 trial, we're only getting to like 7 to 8% mutation. So, can current strategies induce high rates of somatic hypermutation? No. Is the rate of hypermutation affected by B-cell dysfunction or hyperimmune activation? Probably. What factors influence somatic hypermutation? Structure of the target, development and characteristics of the B-cell memory, and there's some data coming out of the Newson-Vide lab to show that induction of CD4 T-cell help is probably vital in creating a lot of somatic hypermutation. So in some work that we've been doing with Susan, who works in Dr. Fauci's lab, is that we've been looking at what phenotypes prevail during HIV infection. And binding antibodies arise within weeks of infection, we know that. And so in the coming experiments, we'll see that we look specifically at chemokine co-receptor binding, and these are non-neutralizing antibodies. Now, 10 to 30% of individuals produce neutralizing antibody after several years. For example, CD4 binding site, broadly neutralizing antibodies such as VRCO1. And they take a long time to develop. So can we gain insights into B cells that elicit HIV neutralization, HIV neutralizing antibodies in the background of initial and continuing B cell dysfunction? And this does have implications for a vaccine that would elicit neutralizing antibodies. So for this, Susan used 3 proteins. She used this is YU2 clade B proteins. She used GP140 wild type. Then she used a GP140 that's knocked out at the CD4 binding site. And she used a GP140 that was knocked out at the co-receptor binding site. So this is non-neutralizing, potentially neutralizing, and GP140. And what she found by looking in untreated individuals was that there was a very high rate initially of B cells that were specific for GP140, which dropped off towards the chronic stage. There was quite high frequency of non-neutralizing antibodies in the early stage, which also dropped off towards the chronic. However, there wasn't a fabulous recognition of the CD4 binding site. And if we have a look at, and then what the proteins allow us to do is to gate on the cells that specifically recognize the CD4 binding site and also the non-neutralizing, the co-receptor binding site. And it allows us to use our friends CD27 and CD21. Now Susan, you'll notice if you've been paying attention, Susan flips these around. So I have CD21 here and CD27 here, and she flips it around. So, but the tissue-like memory remains in the same place, as it's CD21 and CD27 negative. And what we see, there's a larger proportion of those cells that recognize the CD4 binding site, the larger proportion of those fall into the tissue-like or exhausted B-cell responses. Whereas not so many are seen that directed towards the rest of the epitopes that are on GP140, or those that are directed directly towards the non-neutralizing the co-receptor binding site. If we look at the responses exhibited, the activated or tissue-like and exhausted memory B cells in HIV untreated individuals, here, this is represented, and here we have this is activated memory, tissue-like memory, resting memory, and naive. We see that typically, as we've described before, there's a very low level of naive compared to, say, tetanus in healthy controls. There's a large percentage of those directed towards GP140 fall into this activated memory type, and remember that these have got high CD95, so they're moving towards apoptosis. The tissue-like memory cells that are full of inhibitory factors, and then a smaller percentage fall into the target population, which is the resting memory population. As you can see, in something like tetanus, 81% are in the resting population. And then if you put the individuals on antiretroviral therapy, you see that you restore this resting memory population very nicely. So in summary of this, HIV-specific B cells were evaluated by flow cytometry using GP140 probes. HIV-specific subsets are enriched with abnormal subsets that are clearly distinct from other responses. For example, with tetanus, we also used influenza for this as well, and we found the same results for influenza. HIV-specific responses against neutralizing epitopes, such as the CD4 binding site, are slow to appear, and they seem to be very infrequent. However, the responses normalize with antiretroviral treatment and decreased HIV viremia and immune activation. So I'm nearly finished, and I'm going to leave you some final thoughts. So B-cell dysfunction in early HIV infection could have implications for in HIV vaccine strategies that solely target elicitation of neutralizing antibody responses. That is a vaccine strategy that has no effective CD8 T cells. May see the pendulum swing back to CD8 T cells as well. It's important that we can define some of the molecular mechanisms that underlie B-cell dysfunction that may contribute to high somatic hypermutation in the lymph nodes over the course of HIV infection, such as those that we pointed out in some of the in some of the Fluidigm, the gene expression experiments that we've been doing, such as CXCR5, CCR7, and CD21. But we need more evidence from both the periphery and the lymph node from HIV infected individuals. So if we just go back to the schematic I presented earlier, and it's actually been documented that in this compartment, in the light zone, you require CD21, it has to be high, CCR7 is low, and CXCR5 is high. And then to move to the dark zone, CCR7 high and CXCR5 high. So we found those that are actually being perturbed in the periphery. So now what the next step is, clearly, is to go back and look in the lymph nodes of HIV-infected individuals. So, just to end with, I'd like to acknowledge Adam particularly, Sandeep and Madhu, who work in my team, and these, Stephen, Kipper, Marissa and Danielle have contributed really well, but they've moved on to different things now. Rick, of course, in his immunology lab has been absolutely invaluable. The human immunology lab with Rebecca and John Muscola, the University of Amsterdam, and our colleagues over in Dr. Fauci's team. So thank you very much.